Good morning, good afternoon, everyone. So we are resuming our series of meetings to finish up the data release paper. So I have the agenda for today. As usual, this meeting will be recorded, and so we plan to post on YouTube. So if you want us to edit out something during the course of the meeting, please reach out before the end of the day. I thought it would be helpful to sort of go briefly where we are in this data release paper before we dive into this. So first, well, the review piece came back from review, from physiological review. So we are planned to discuss that with all current co-authors of the manuscript on June 22nd, next Monday morning. And next week we'll have a meeting with Richard and Christophe that will present a potential data set that we plan to collect on EEG data in humans. So this is just the overall context. Today we plan to discuss receptor field, which is one of the figure that we plan to do for the data release paper, which I have here. So currently, the majority of the methods is written. And I also iterated the first few figures of the manuscript. Given that the reviewers for the review paper are pointing out that we should shorten the experimental proposal, I think it's likely that some content from the review will come in here, which is fine. But we'll see what the discussion says next week. I edited this figure here, but you see, let's see if I can open it up. But sort of introduced the experimental design. I just added a panel in C and edited D to contain all of the stimulus blocks. So it was updated because our previous version did not contain zebra and receptor field, but we are talking about today. So that should be clearer for everyone to understand how the stimulus was constructed. So it contains the different control blocks that we presented to the animals for all three modalities, as well as the different context with a color labeling here. And then the durations of the different block that were part of the experimental design. So this was changed. And then we also are working towards this figure, which is the overall design. And so the rig geometry was added. This figure that highlighted the targeting geometry is, I think, almost finalized. And we are currently working with each team at the Institute to finish up this last column. Marcel, you have a question? Or?
Yeah, just I cannot see your screen.
Oh, you cannot?
I can't. But maybe it's me.
I can, yeah.
Okay. Okay, I will figure it out.
Okay, I'm sorry you have issues. Yeah, so this is the two figures that are sort of introduced to experimental design. And the following ones are intended to introduce the data. So we were planning to have a figure on SNR. Yeah, but it should be a figure 4. It's highlighted here, but we still need to finalize. And a figure on receptor fields, which I think, so the intent is to have this across all three modalities. So to highlight the convergence of the experimental design for 3 different techniques. So we are there. Carter is still working on uploading the latest SLAP2 data. We are seeing receptor field in this. So we did some sanity check. And so you'll likely will get this within the course of the month. And today, I think Lucas and Marcel were planning to present some of their results on EFIS analysis of receptor fields. Any questions on this overall context? Or? Okay, so is it Marcel or Lucas that will lead here?
I will going to present.
Okay. Are you able to sort of share your material?
Yes. Okay, can you see it?
Yes, thank you.
Okay, perfect. Yeah, so this will be an update from our side about the receptor field analysis. And here I will focus on the analysis of the zebra stimulus, because this is something I would assume which is probably kind of new to most of you, given that the stimulus is also very new. So first of all, I will give like a short introduction into what the idea of the analysis pipeline is. So we have the stimulus, which is basically a noise pattern with these moving stripes here. And we want to analyze whether a neuron recorded basically corresponds to specific features into this stimulus. And we know that in mammals, typically the receptor fields are shaped like Gabor wavelets. So what we can do basically is to break down this stimulus into in this Gabor feature space, so to say. So what we do is we define a whole library of different Gabor wavelets, and these wavelets have different properties. So they have a position, so an XY position on the screen. They have an orientation. They have different sizes, and they have different frequencies. And what we can do then is to just convolve the stimulus with all of these different filters. And we get for each position on the screen an activation map. So basically how active on this position on the screen a specific feature, like a specific Gabor wavelet, is given the stimulus to a specific time. So here we have 3 different examples. Here, for example, like we have the first one in this subfigure D would be like a horizontal filter that basically filters out all of these horizontal frequencies or spatial frequencies at these different positions. Like the other 2 filters presented here, but with different orientations. And what we can do now is to look at a specific position on the screen. So for example, we have this wavelet, we take this filter here, and we get this activation map that is on the same position on the screen. And we get basically a signal over time on this position on the screen on how active this filter given the stimulus is. And of course, so the filter stays constant, but the stimulus, of course, changes over time. So we get this activation amplitude. Don't get confused that this amplitude here is only positive. That's just an example from this paper here. In general, the amplitude can be negative or positive. And then we have from the EFIS recording, we have a units firing rate. And all we do is basically computing the correlation between these 2 signals. And then we get basically how a neuron correlates to a specific Gabor wavelet. And we do this for all kinds of different wavelets and get then in the end correlation maps.
Marcel?
Yeah.
Sarah has a question in the chat. She asks, just to clarify, the zebra stimulus uses one overall speed, but is fixed to transition across time.
Yes, that is correct. So the overall speed of the stimulus is the same. If you give me like just a few seconds, I can show you an example of the stimulus. So I think you cannot see it, so I have to reshare. So now I hope you can see the whole screen basically.
I cannot.
And maybe so don't look too close onto the stimulus. Yeah.
Well, we can't see it.
You cannot see it.
Yeah, we can see it.
Okay, now. Okay.
That's right.
Yes.
So here in the middle, you can see. Hopefully, I don't know how the rate is on Teams. So the frame rate. So basically, what you do is like the overall speed of this stimulus is defined constant, but you have all kinds of different temporal and spatial frequencies in there. So I hope this answered the question.
I think it should. Sarah, feel free to put the comment in the chat if it doesn't.
Yeah. So what we do right now is only so we don't map the tuning in time. So temporal frequency, that's correct. But in principle, this is something you can more or less do easily also with the stimulus. Yeah. Good. Okay. So we get these two signals, and all we do is basically compute the correlation between both of them. And what typical, okay, and another thing, so you can do this for different kinds of wavelets. So I dropped like the last property of these wavelets. They cannot only have a frequency, but also a phase, of course. And what you typically have is you take sine and cosine phases. And then there are different ways on how to keep on. So you can analyze these different phases separately. This is something like this approach I will show here. Or what you can also do, you can combine both phases by just summing the square of both activation maps. And this is a result, this is an example shown here in the last row, basically. But typical results for this presentation will look the following. So here on the left side, I will plot the so-called receptive fields, or what we call receptive fields. So these are all the positions on the screen. And what we do is basically we find a filter that is like the best correlating filter to a unit's activity. This I'm showing here. And how you have to read this left-hand figure is basically when I take this filter and I do all of these convolutions, I go a slide back. So I have this filter here, and I do not only do like the activation, or I take the activation from the filter from this position, but on all kinds of position. So this is basically the activation map would be this in the second row here. And what I'm showing here is basically when I take this filter and I put it on this position here, what would be the correlation value at this position between the activation of this stimulus and the units firing. And here you can see like the blue color indicates that it's a negative correlation. So I'm showing this for both trials. So we have two trials of this kind of stimulus right behind of each other. And we can see, okay, these what are called receptive fields. So these are basically correlation maps are kind of similar. And also like the best filter we found is also kind of similar. Here I'm showing the phase 0. And of course, we can also do so we have the sine and the cosine, or here it's the cosine and the sine. If I do this with the phase p half, so the second kind of filter we can use, we get these second, let's say, variant of a receptive field. And here, I just want to make the point that how we can make sense of this. So basically, when I have this filter here, the phase 0 filter is always very similar to like where the frequency is very low. It's more or less like a single bump, more or less like a kind of similar to a normal distribution. So we have this Gaussian bump basically, and we filter the stimulus with this Gaussian bump, and we get this negative correlation here. And what we can do now, when you think of, okay, I have this p half filter, and now I'm basically, when I move the p half filter a little bit above this Gaussian bump, then I have an overlap of this negative phase or this negative part of the filter with the bump.
Is your slide already showing the
You don't see the slide? Oh, that's a problem.
Well, okay, now we somehow at least I didn't have it on the screen yet.
Okay, can you see it now?
Yes, now it's there.
Okay. Was it the case for everybody? Because then I have the last 2 minutes.
No, it's somehow we were missing the phase P2 columns.
I think you went, we had presentation mode and you had the slides, maybe not in presentation mode or something like this. I don't know. Anyway,
okay.
Now it's clear, I guess.
Okay, then I just repeat the last part maybe. So here, like we have this negative bump, yeah, coming from this positive filter here. And when you think of, okay, we take now this p half filter and we move it like a little bit above this bump, then we have an overlap of this negative part of the filter with this bump here, giving like a positive correlation of this filter on this position. And if we move it like a little bit below this bump here, then we have an overlay of this positive part of the filter with this negative bump here, giving a negative correlation. So I hope it might be like on the first view make a bit confusing. I hope it's more or less clear, because like this behavior we will now see on all kinds of different examples. And I want to explain another last example. So what happens if we find receptive fields with higher frequencies? So here we have this phase 0 filter with a little bit of a higher frequency than in this first example, giving like the following activation or correlation maps. So basically we can see this neuron here is becomes very active when like it sees this kind of pattern on the left hand side of the screen, but is still like a little bit active when this pattern occurs on the remaining part of the screen. And when we see like the phase shifted filter, or we do the same testing with the phase shifted filter, we see like a phase shifted version of this activation of this unit, which still makes sense. So this is like this slide is only like to make a little bit of a reasoning that I think in the end, like the results, we can make sense of the results and why we see different patterns when we take different filters. Okay, so So what we do, we have these two kinds of filters, so these two different phases. And what we can then do now is further optimization. So we do not only optimize these different filters, but we can also optimize like in which time window we are computing the correlation between the activation of specific filters and the unit's activity. So, and like these windows have two properties. There's a so-called duration. So it's basically like how long is the period of spikes I'm counting basically after I present this, after a given frame of the zebra stimulus, for example. So this would be the duration, and we have a delay. So what I would assume is that basically a signal needs a little bit of time to, like, when we present it on the screen until it shows some activity on a specific brain region. So we can also optimize these delays. And what we typically would expect is that we find some short delays with some, let's say, medium scaled durations.
So this is the window during which you integrate the response. Is that right?
Yes,
Okay,
exactly.
that makes sense. It's very clean.
Okay, and we have to do this for each unit because potentially each unit could integrate over a slightly different time window. Okay, so let's go to the main part and have a look on our best receptor fields. Here I'm showing probe C. So this aims to be located on V1. And the first row is basically the row I've already shown you. And the other rows are so the order of these of this plot is basically when you that it's ordered by the absolute maximum amount of correlation I'm finding. So it's basically the peak value. So when you go down in this plot, like the amplitude of the correlation I'm finding goes a little bit lower and lower. And what we can see here is that more or less, I would say these top receptive fields are kind of similar. So they are kind of small in size, at least most of them, and we can see very small values for delays.
But
then
you do see large ones, right?
Yeah, there's here in this example, like there's a large one, yeah. Like a pretty large one actually, like exceeding more or less like half of the screen.
And this is only in V1?
Yes, this is, I mean, like it's from probe C, and I think like the histology is not in the data yet, but at least it aimed for V1.
It's amazing.
There could be thalamus on the tip, right?
Yeah, yeah.
I'm not percent sure. Severin might know or will know. But
yeah.
So we didn't. It's just all probe C. So that it could be subcortical.
Yeah, so one could also include the depth, of course, here in this kind of analysis, but I thought I can just wait until maybe the histology is there.
You could be looking at the VIP neurons for some reason.
Would it be possible that it's an LP cell? Like maybe with going through V1, we ended up in LP. That's we will know soon. So just to update you, like there's only 4 brain that we still have to annotate and everything, but we are nearly ready to give you the CCF. We just have a little technical thing before we can share it. But like I would bet that this big receptor field is not V1 for sure.
Yeah. So CCF would be the histology then?
Yes.
Okay, yeah, that would be amazing. Yeah.
Yes.
That would be great.
Yes.
Yeah. So so far I do not really distinguish between different areas because like it was not in the data, and then I found it a bit arbitrary to include some depth filtering or so. So this is for probe C, and I do this also. I do this kind of analysis for all probes, but here I'm showing also probe B and D, which also should be targeting visual areas.
Just a comment. We should thank Sophie to has pushed us to double the number of zero because it's clearly helpful here.
Yeah. Like from this overview, I think there are a few messages you can take from this. So first of all, I want to highlight maybe probe D, because here you can see clearly like delays, which is great. I will show another example in the next slide, showing some problems I have also with the delays, but here it looks fantastic, I would say. Another outcome from this kind of analysis is that both trials are very similar. So we see like very similar amount of here absolute correlation values between both trials. And the most difference comes whether we look at specific phases, let's say. Don't get confused by this. By this data point here on the bottom left. Here it seems there are some data points missing in between, like this low delay value. And I'm sorry that actually, like for some reason, Google Slides don't show the whole slide. So here, this left column is the duration, and here the right column is the delay. I'm sorry, it's a bit cut off here.
Just a quick question. I noticed that sometimes between the trial 1 and trial 2, the orientation preference seems to shift 180 degrees. You see that?
Yes.
Like so that probably means that the vertical, or the vertical up or vertical down mode are very close to one another, right? In response, and when you repeat, do you see what I mean?
Yeah. Oh, that's actually like a good interpretation of the data. Yeah. But I'm not so sure, to be honest, because the stimulus should be very diverse. So the stimulus itself doesn't change, right?
It's the same movie that repeats twice.
It's the same movie, yeah. So, but can you point to a specific example right now?
So for instance, if you look at the row number 4 on the right, probe D.
Yes.
You see that between trial 1 and trial 2, the phase 2 seems to shift up down.
Yeah. So I would interpret it as the following. So in the end, this unit will have like a real receptive field, and I don't expect it to change it so quickly. So what I would assume here is that like with what I mean with real receptive field is that it only can have one phase. And here I'm just basically from these different phase shifts I could analyze. I only analyze here 2, but it could be everything in the middle between them. And here I would say the real receptive field should be closer to the phase 0, because when you have a phase 0 activation, you see this, of course, when you filter it with a phase 0 filter, but you can also get an activation from the p-half filter, but with a slightly off shift, like in the direction of the direction of the frequency. So let's say. So that's something.
Isn't p-half telling you about the orientation tuning of the unit?
Yes. I mean, you also have the orientation. You have the orientation in all of these filters, but the orientation becomes more and more important when you go to higher frequencies. So when you are at a very low frequency, like in this example here, you don't see a big negative part. So there's not really a very active frequency tuning here. But when you go to higher frequency, you get these negative parts in. What I want to say is that basically it makes sense, at least to me, that when like the neuron really correlates with this phase zero filter, you also get a result for the p half shifted filter. Whereas when it would be the other, like the opposite, the other way around, then we should see like a phase zero positive activation. Let me think for a minute. So you would still see a signal in the phase zero in the opposite case, but I would expect it to be much lower.
Yeah, and we can also see that the phase zero has overall much higher correlation, like in the duration plot, for example. See that nicely. So it's essentially the phase two is just trying to approximate the phase zero as best as it can. And then all it matters is where it puts the, let's say, the positive or the negative part, and then you can turn the other one around. It doesn't matter, so to speak. So it's unconstrained by the response of the neuron. And
And now coming back to your original observation, Jerome, when you take like this, you take this negative bump here in the middle, then you can overlay it with this p half shifted filter in two ways. So first of all, you could, when you have this negative bump here on top of the filter, right? You can just like place it slightly below this negative bump here, and you have an overlay of this blue with that blue, right? Like returning a positive correlation on this bottom half. And if you would do it the other way around, so you take this filter, you move it slightly top of this negative bump here, you have an overlay of the blue and the red, resulting in a negative correlation. And of course, you can also do this with a 180 degree shifted version. And then you get basically the same is happening, but you get like one or the other kind, let's say.
Sorry, did you ever find a unit where the phase two is really improving, let's say having higher correlation? Does that would mean that unit really has a receptor field where you have one positive or one negative part, whereas the cosine is you have like this bar tuning, right? You have one bar and then you have this suppression, but symmetric on both sides.
Yeah. So I cannot really remember to saw a filter like that. I could just test it, whether in some cases I find higher correlations for this shifted phase. Another way one could do it is basically not only test these two different phases, but really optimize the phase. But one of the like outcomes of my experience with this kind of pipeline is that it's very expensive, and you don't want to grow this amount of tested features too much. Yeah, but one possibly could do this if this is interesting. I will come back to that later. Next, I want to show like the same graph, but for another session. So everything stays the same, but here's another session. I think I have maybe except of probe B. For some reason, probe B stays the same, but probe C and D are different. And here also you can see that on probe D, we don't see this clear delays anymore, which to me is a bit surprising. Yeah. because overall, let's say we should find a very small delay. The delay should never be 0, but for some reason here in this session we don't see it so clearly. And there are two, Lucas had an idea how we could explain this, because in the end, like the stimulus is very, very predictable, and of course you have a lot of activation like going on even in V1. So it could be that maybe this highly predictable stimulus just gets like more or less explained away. And the feed-forward input from the retina is maybe not too much important in this case. I'm not very sure about this. I think one should still see like a maximum amount of correlation when actually the signal from the retina enters this specific brain region. But at least it would explain why we see like a non-zero or a positive amount of correlation at 0 delays.
I mean, if that proposal, I wonder if you could look at, because we represent Boost stimuli 4 times in a row. And so if Lucas is correct, the delay should decrease, or, you know, as the mouse experience at zebra more and more.
Yeah, that would be an interesting way to test it actually. Like we could chop the stimulus in smaller parts and see.
You could do that, but there's also like there's also 4 consecutive sessions where the mouse is experiencing zebra.
Yeah.
It considers that these correlations, it's essentially like moving edges, right? So that's a very, how to say, universal feature, I would say, of world statistics. So I would assume it's really baked into V1, like from this, or at least, you know, in these mice, it's probably in there already. That essentially, when you have like a moving feature, you know, adjacent V1 cells, let's say, already activated, and this is probably, I would say, it's kind of a primitive feature that's in there already. But yeah, this is just one idea, but it's true. I mean, this 0 delay would imply that within 80 milliseconds we see most of the response, which is quite fast if we compare it, for example, with these oddball response curves or so. Yeah.
Sarah is making a comment. Maybe there is adaptation to the movie, which I think this is what we're talking about. Yeah,
exactly. Okay, what we can do now is like we have this kind of different delays, and we have all of these optimized filters, and we can do a little bit of a meta-analysis of that. So basically, we can have a look on the mean delays and the receptive field sizes for different probes. And here I'm showing for like the amount of normalized absolute max correlation for different delays. And we see like for probe C and probe B, we get like the maximum is at very, very low values. It's exactly what I talked about. That here for that session, like we see this very, very low delays, but clearly for probe D, which should aim higher visual areas, we see like a shifted delay. So that's basically what I wanted to test here. To see, do we see different delays for different probes? And I think that hopefully becomes more clearer when we have access to the specific brain regions for the specific units. We see this. What puzzles us a little bit is like this second peak we see here on the results on probe B.
Marcel, did you look at the frontal probes at all?
Yeah. You would see examples from the other probes in I think the next slide.
Okay.
I just here focus on the visual, like the ones that
That's right.
aim to the visual areas.
I'm not going to steal your thunder.
Yeah. So here I'm showing the same plot, but here for the different receptive field sizes. So it's basically the size of the optimal fitted wavelet. And we see here that for the probe that aimed for V1, we see much lower receptive fields sizes than for the others on average. Here, this is like a more or less very hands-on analysis. One maybe have to rethink how to compute the critical intervals here or the standard deviation. So here I use the critical intervals. Maybe it's not the best choice for drawing error bars, but yeah, don't get confused too much by that. I think here the mean tells the very stories. Okay, so this is just to. So in the end, I would summarize that I think this kind of pipeline makes sense so far.
Can I ask you just a meta question? Do you feel like Zebra is already providing more information than the Gabors?
I will show a comparison.
Okay.
In the second next slide.
I keep asking those questions.
Yeah. But next, I want to I wanted to have a look on the average receptive fields, and for this I have this figure. So here I'm showing all the probes. So probe A to F, and what I'm doing is basically I average all of the correlation maps I'm finding. And when you focus here on the first row, where basically sum over all the units we have, we see basically not a lot because everything averages out, which is already, I would say, a nice finding because this is something we would expect. But when we filter by units that have a higher correlation to specific specific features, we see a more and more clearer evolving average receptive field. So there are questions. I think he showed the delay one. Okay, should we jump back to the delay part? We do not expect.
Don't worry, Marcel. Lucas responded.
Okay, so
yeah, I guess that you mentioned it. I missed.
We don't expect a higher delay for this P.
I think part of the confusion is the order of areas in the axis.
Yes, that really confuses things for sure. So I would recommend to start with VSP.
It's ordered alphabetically after probe.
Yeah,
I'm sorry.
We have a hierarchical ordering in previous manuscript that you could use to position the area. I think that might be less confusing.
Yeah.
But even with that orange on the left side, that should have the shortest delay. And I think that's what Lucas wrote to me, that you guys are also confused by that. So on the left panel, orange.
Yeah. Yes. And it has a very low delay, or am I wrong?
Well, but with A seems to have lower delay than P.
I mean, the critical intervals are kind of large, so I would not really distinguish between both of them for very low. So at least for with P and with A, I would not really.
Okay, I thought you were making a point about the different sizes and the delays, but
yeah, I guess one is more, we need more data or put more sessions through, and the other point is that these delays for this A, they have this kind of second peak of the delay, so we're a bit
right. And this is all computed based on the Zebra stimulus?
Yeah.
And from which sessions?
The session is here on always stated somewhere on the slide.
But these are the sessions that we discussed for the paper, Jerome? Or are they like, right?
Yeah.
Okay.
I mean, I don't think we decided to focus on specific subset of session for the paper. I think Marcel and Lucas can decide to focus on example from any of those.
So what I basically did is I went through the notes of the different sessions and I picked ones that basically had the best like detailed description, like there was no iPhone foam. Their like out probes were going well, like there were no overall problems. I selected these sessions.
Makes sense. I mean, Zebra is at the very end of the session, so you're more sensitive to accumulation of stress.
And also, like one of these sessions, I think also Alexander Meyer analyzed. So I have like this. Sorry, I have like this here with the like the 830794 and the 95. And I think one of them also Alexander analyzed.
Sarah has a question for you.
In the chat.
Yeah, she said, "Did you select tune cells or only for the delay?" So I think she's worried that untuned neurons might impact your measurement.
Yeah, that's a very good comment. I didn't do this here for this plot. Yeah, I should have done that.
You should explain why you have very large STD bars.
Yeah. That's a good comment, because like coming back to that slide here, I did it here. I should also do it, of course, for the receptive field sizes and the delay. Yeah. But what I want to say with this overview here is that basically it makes sense to define like a threshold, a correlation threshold, because otherwise like correlations would average out, which I would say makes sense, and that we also see here some higher some kind of receptor fields in other areas. So also on probe B, E and F, but with higher frequencies. I have the same plot for the other session, and it looks like this. And here the same year also we find results on probe A and F additionally with higher frequencies. Don't worry about like this stripes here. This is basically due to like a low number of significant units we find here, and then like it doesn't average out so nicely. And something went wrong with my results file here on probe B, but I also have results for probe B. Yeah.
Marcel, I think the slide skip was a bit delayed or so. Can you just quickly walk us through the rows and columns again?
Okay, yeah. I will jump back to this slide here. So basically each row shows results for a different probe, and I average all the correlation maps I find on this specific probe. And then I introduce a threshold, and this threshold basically caps or throws out correlation maps that are that have a maximum below a given value, and these are here the rows. So here on the first row, I'm just averaging everything I find, and we see like most of it just averages out. And when I introduce max correlation threshold, then I sort out some non-significant units, and then we see a more and more clearer emerging average receptive field.
I see.
Also, like I hope you can read it here in these inside these plots. The n equals is basically the number of units included in this plot. So here, for example, like in the when I only have one unit that have a correlation above 40%, which is actually a very high value, I find. But on other probes, you find like a little bit higher number of units with these high correlation values. Okay. So this would be one way of one could average receptive fields and show an average receptive field for different areas, for example. And also like here we can see like the two different trials are very similar. So in the end, one would also average between trials, of course. Okay. Now I want to move to the comparison to the analysis with the local Gabor patches. And these figures will look the following. Here I have like the right part is something I actually already shown. So we have like the correlation maps. Here these numbers indicate for the zebras like the maximum absolute correlation value I'm finding. And here, like in the best filters, I'm also showing the theta, so the orientation of the filter. And on the left-hand side of the figure, I'm showing the activations of the local Gabor patches. So each pixel here is basically the z-scored difference in firing when I'm showing a Gabor filter on this specific position with a specific orientation. And here the numbers, I hope you can read it. I plotted on the GABA receptor fields are the rounded p-values I can compute for these findings. Don't be confused, like the real p-values are not 0, but they are rounded to 0.00, but they are not really 0. And what I can do now is basically I can select like the most significant GABA receptor fields, and I draw for the same units the zebra receptor fields, and this is shown in this figure here. The most important thing I think I would get out of that here is that we have a nice alignment in these examples regarding the position of the receptor field. So here, maybe I should remind that the local Gabor patches are not spread over the whole screen, but only like of the middle part, I think from -60 to +60 degrees. And the zebra stimulus utilizes the whole screen, so it's basically a little bit wider, and that's why here the aspect ratio is a bit different. So basically, like if you want to overlay it, you have to overlay this GABA receptor field at the center of the zebra correlation map here.
Sarah is pointing out, not sure if you did this, but maybe it would be useful to shift the unit along X and Y to center them based on the peak response position. I think she's just sort of highlighting if you don't care about the XY position and want to see the shape of the receptor field overall.
Like to test whether the shape of the receptor field, for example, or its size, for example, is similar.
Yeah, I mean, just if you want to, yeah, it depends what you want to focus on in your plots.
Yeah. So what I here was interested in was actually the position. So the position also should be similar.
That's right.
And for me, it looks that it actually makes sense in most of these cases. Also, what I think maybe makes sense is when you have like more like a higher frequency and more like diverse correlation map for the zebras coming out, then also like the activation to the different local GABA patches is a bit more diverse. Yeah, but overall, I would say this is like I was kind of happy to see that because roughly it was what I expected. But of course, both are different stimuli. So the local GABA patches are static, and the receptor field sizes are dynamic. So you would expect at least some differences. And this you can see when I did a different selection criterion. So what I did here, just to repeat myself, is like I selected the most significant GABA results and matched the zebra results for the same units. I can also do the opposite around. I can take the most significant or the zebra results with the highest amount of correlation and plot the corresponding GABA patches. And these results are not I mean, they're still like Yeah, actually, in this example, now when I look at it, it still makes sense. But what I see, like when I look on more and more units, that basically you see more examples like here in this, like these two here. So basically that you have a very locally defined response to the Gabor, to the zebra stimulus, but a very diverse response compared to the local Gabor patches. And in these cases here, like these results are not even significant. So here I get p-values of 44%. So this is not even like so what I want to say, is that we can get very like highly correlation between a unit's activity and a zebra stimulus, and still find non-significant results for the Gabor, or at least for some Gabors. Sorry, so these results are relatively fresh. So while I'm speaking, I have to correct myself a little bit. So you have to see, like, you see very high p-values for the 0 degrees. I'm sorry for the confusion, but actually here for the 45 and 90 degrees, you get higher amount of p, so more significant results actually. But still, like the activation is more diverse, so it's not so localized.
Marcel, do you have many more slides to show, or?
No, I'm actually at the end. So I
Okay.
only have some backup slides if you want to have a look on like specific receptive fields for single
Can I
units.
ask you a quick question, Marcel? So can you summarize then what is the difference between Gabor and zebra? Like, what did you learn at the end?
Yeah, okay. So I would say it really depends on what you're interested in. If you're interested whether there are receptive fields and whether the receptive fields are roughly on the screen, for example, something you probably want to show for this paper, I think both are fine. Maybe the zebra is even a little bit better because you get a little bit more extra out of it. So the stimulus is much more diverse, and because of that, you get all kinds of, for example, different tunings out of that. Something I didn't show here is that you can really also get the tuning curves out of that, which is kind of nice. Given that also this stimulus takes less time than the local Gabor patches, and you can basically get more out of it, right now, if I would have make a choice, I would choose the zebra stimulus.
Can we step back a little bit? I feel like so for the purpose of the data release paper, I think like a subset of this analysis is probably irrelevant. I think like you could take some of this effort and show that you have reliable responses to both the Gabor and the zebra, and show those across the probes and across the areas, and that would essentially be the figure, right? And then I feel like you're already sort of shaping a follow-up studies here. Like I feel like you could dive much deeper into the comparison of the two stimuli and sort of highlight the richness of the zebra responses across areas, and still start to make hypotheses as to what support these. But for the data release paper, I think we just want to highlight that you are empowered to do this analysis. And so I wonder what would be the nice next step there.
Yeah, I also wanted to discuss this. I'm sorry that I took so much time.
No worries. It's amazing. I think you have lots of interesting results. Alex, do you have thoughts there?
Yeah, so first of all, that was really great, very interesting. I loved that you went so slowly. I could understand everything, and I appreciate all the detail. So I'm thinking maybe we could have both. You could show matching results for the gradings and the zebra, And then also show, because it's interesting, that what you have right here, where the zebra might be more superior. So coming from this tradition of receptive feed matting with gradings, we know that there are problems with center surround suppression, which could exactly explain this right here, that some receptor fields, you don't see them, because with a grading, you also stimulate the extra classical surround, and you might suppress the neuron. But on the flip side, zebra makes a lot of model assumptions, which each of them I could question, and you could get spurious results, right? So you could ask questions about statistical power, about using these filters and how much they apply, the question of linearity, non-linearity. So it's actually an interesting question. Then can I trust the modeling? Maybe it's a problem with the modeling, or can I trust the gradings? And so we could show both here.
So I would try to think of creating one or two figures that sort of is enough to get people interested in the data, as Alex pointed out, and then we could put this in the manuscript, and then you could continue in your own paper to dive deeper. I think that would be my intuition.
Maybe we could, I don't know if you have still 2-3 minutes, because that would help us a lot to move on. Maybe we could pull up this slide, this figure from the paper, the data release paper, because there we drafted a bit an outline of what things we plan to include. We have it, Marcel, as Jerome, I think.
Yep. Give me a second. You mean this?
Yeah.
Yes.
That's great.
Because so far, what we thought is that we might have essentially like an averaged version. That's the one in, like, if we go into the receptive fields row, so like the averaged one between the patches and zebra noise, and then we have example units. Maybe that's enough to have, like, if we select interesting units, we can discuss the differences, and then in the averages we can show that maybe, I don't know, zebra noise. For example, I found this in the LM really interesting, that you had like a very biased sampling of that thing along this. It's not shown here, but it could be interesting to have the averages and then the individual units. Or we just have one figure dedicated only to the receptor fields, and then we could show more. But yeah, that's the question. What's the scope for the receptive fields? How much space do you want to give it?
So do you want to show examples only, or also some kind of population data?
Yeah, so the idea would be both, so that we have like the big one. I mean, now it's just like placeholders, but the big ones would be kind of averaged maybe for a single session, to just because the probes, it matters, how they sample this. So for one session, the average over all the units, maybe with some cutoff on the correlation values, as was explained, and then we could have interesting units next to it. Have example cases where I can discuss quickly how these approaches differ. So, and then of course cross model.
Do you think you could try out to do a similar analysis on the mesoscope data for zebra?
Yeah, so that always was the idea. In theory, I only have to exchange like the input signal instead of spike rates you put in whatever. I hope like getting the signal out of the mesoscope, I hope it's not too difficult. I simply had not the time for that yet, but it always was the plan.
Because in the mesoscope, you're going to have a rich population of inhibitory neurons, and so the diversity of receptor field might be larger.
I have a quick question. So because we also in the NeuroPixels data have different areas, should we stick to this figure using V1 data only so that it matches for the 3 technique? Okay.
I think that could make sense.
I mean, in the end, I think one problem will be that the figure can end up to be too much cluttered. I think one has to make a compromise somewhere, and probably like cutting down areas is one of the easiest, maybe.
I think I would suggest to just do V1 for this figure. And then maybe you want to add a small supplemental that dive deeper into the neural pixels so that we can say in the text, we see a rich diversity receptor field in different areas, including in the ACA, and just sort of highlight, and then the reader can go there and see, oh, that data is really rich in that sense. But
yeah, another opportunity. I mean, if they're supplementary, I think I would totally love to do that. And another opportunity maybe would be just like if we also want to provide like these analysis scripts and so on, and there could be also like a bunch of notebooks for different specific interests, for example. But of course, it would be nicer to have it in the paper itself.
Yeah.
Okay, can I ask a very naive and possibly for most people here, stupid question? My training was not V1, so I'm not a V1 person. So I'm very curious, like when we keep repeating like comparison of these receptive fields. So we show the receptive field, a small patch at part of the visual field, right? Some neurons respond. This means that these neurons are encoding information from this part of the visual field. Good. Now, are you saying that depending on whether this is a Gabor patch or a moving gradient or a zebra noise, we may get different sets of neurons that encode that part of the visual field? Is that the point here?
I mean, I think there's overlap. So, like, I think one problem with these kind of analysis is that you never can show or test all different kinds of features a neuron could potentially encode. And for example, what I skipped here totally in my kind of analysis is the temporal aspect, for example, which I think when we want to compare it to the Gabor filters is not very dramatic because the Gabor filters also are static. But of course, in the end, the zebra stimulus is dynamic. So also, and in the end, it's also like, by principle, it's not very problematic to also include the temporal aspect into this kind of pipeline for the zebra stimulus. But in the end, when you want to you want to get an idea of what maybe a neuron could encode for. And with this kind of analysis, I would say you get the most prominent features.
Like
but in the end, it always will be a combination of a whole bunch of different things.
Right. So the idea would be to know as much as possible about the different encoding properties of these neurons and to have a very rich receptive field stimulus that can extract all of those. And the idea is that zebra can give us temporal information, spatial, and like different tuning. See, but it's not that we get different sets of neurons. Like it's not that one neuron. If we show the receptive field on top right corner, whether depending on whether it's zebra or moving gradient or Gabor, we'll get different sets of neurons.
So what I show on this slide here, these are the same units. So
but the sorting is different.
Sorry?
I thought the sorting is different between left and right.
Yeah, it's just how I select the different examples I want to plot here.
Okay, but it's the same. Each row is the same neuron.
Yeah. So maybe so each of these rows here. So each of these here are the same unit.
I mean, I feel like the intent, those stimuli is system identification. So we have a blocks of stimuli where there's adaptation and learning that occurs through the session, and then at the end we have the system identification blocks that allow to characterize the baseline properties of the cell. So the fact that they're really rich is really helpful.
Right. Okay, thank you.
It's 10:12. I want to be mindful of the time. So Marcel and Lucas, are you gonna try to sort of consolidate a V1 focus figure? I think that would be helpful.
Yeah, and also for the mesoscope then.
That's right. And then try to do the same V1 focus on the mesoscope. I think that would allow us to make that panels.
And then SLAP, is that on the way? Will we also have CPRA data for
That's right.
SLAP?
That's right. Carter, do you have any update there?
For SLAP 2 NWBs?
Yep.
Question? Yeah, we are close. There's actually some infrastructural issues that are out of my hands blocking us right now. So we're waiting on some people at Code Ocean and some of our infrastructure to be able to fix a problem. And then I will be able to test. I have the NWB packaging script all done, but I need to test it and validate all the outputs when that's ready.
Carter, can I ask a quick follow-up? So I tried to look at the really interesting thing would be having all the inputs, right? The single vesicle, glutamate vesicle imaging, but then also getting like a 2-photon kind of output signal, even if that's only 20 minutes because of photo bleaching. And so unless I did a mistake, I couldn't see it in the NWB for now, that 2-photon signal. So will that be part of the new release also? So we could look at input, output for one run?
I mean, the whole actually, question for Jerome, I suppose. I mean, it's just the glutamate red and green channels, right?
Yeah, we have the red channels calcium. So yeah, so it should be. Well, this is part of the things we did. So Made in Casper's teams has been annotating all of the files and segmenting the soma, and so we did all of that work. So it's going to be in the files.
Oh yeah, that's a question. Then yes, we have the trace for the soma. Yeah.
Kareem?
Yeah, it's great work. I just have a question regarding the data sets that have the sensory motor condition. Because I asked us to start to look into that. That's part of the things that we are very interested in. So I was wondering what's the status of the curation of the data, and if there's anything similar to what we've been seeing being analyzed in the sensory only condition. Where can we find this information?
Can you clarify? You mean because I mean Marcel looked at the zebra stimuli of a large number of sessions. I don't think he focused on any particular context. Whose sensory motor sessions are uploaded?
So they are there.
Yeah.
Okay.
Yeah. If you look into my postings, Kareem, I looked at the oddball conditions for the sensory motor, and I found good responses in the 2-photon and the mesoscope. NeuroPixels also, there's some interesting differences. That's really just first time pass, not just throwing it all together, but there seem to be responses and they look interesting.
Okay. Thanks, Alex. Appreciate it.
Okay. So next week we'll have a presentation on EEG in humans. So this will be a very different session. And then in the following weeks, we'll have to plan a schedule. I wonder if the 2-photon folks with Farzaneh can provide an update in 2 weeks.
So I know Davis is working on it. I've been also meeting with him to get some in between updates. He is supposed to write on GitHub and propose a date. I don't know if it is going to be next week or the week after. I think he was planning on next. Yeah.
Ideally in 2 weeks.
That's actually better for him. I guess he'll have more time. Yeah, I think 2 weeks from now he will for sure be ready.
2 weeks is good for me.
Okay. So let's do that.
But just to make sure, he's working on the different SNRs because from what we understood, you want it simpler than what we had thought.
That's right.
Were going to, so all you want is basically the FOVs from the different planes and some SNR measures.
That's right. So let's do that on Mesoscope, and then we'll have to translate that work to ePhys as well. So just,
Sounds
yeah. Okay.
But what he had for our lab, and I thought initially that's what you want as well, is more of a like interactive file that we go through them one by one. But from what I understood, you want just something more static, figures, individual traces, SNR, something like that. I guess when he presents, we can have a more in-depth conversation, but
that's right.
That was my understanding.
The SNR figure is to highlight signal quality across the different modalities. And then the follow-up figure that Marcel discussed today is receptive field. So we're starting to look into more system identification kind of things. And then the last figure is oddball response that we haven't discussed too much.
Yeah, yeah. And then for the SNR, so is it like you want individual traces with SNR, or you want the distribution of
individual examples plus distributions?
Okay. Yeah. Individual, good, poor, like with different qualities. Sounds good. Yeah.
That's right.
Okay, thanks.
Okay, so next week we'll discuss the paper on Monday and then the EEG in the next day.
The paper, you mean the review comments?
That's right, the review of the review.
Well, one reviewer made a very interesting comment, is that you need to provide coupons for coffee for people, expect them to read a 99-page review. Did you read that?
Yes, we can provide coupons if they want it.
I'm sure. I mean, that's what he's implying. It's the funniest review comment I've ever seen.
Yeah.
Anyway, yeah, I look forward to that.
Thank you.
Jerome, are you going to be around for just a minute? I just need to talk about next week.
Oh, okay. We can do it a minute. This is still recorded, so if
yeah, so let's. I'll invite you outside of this.
Okay.
Bye.
Thank you.
Bye.
Bye. Thank you, Marcel.